Good evening, 6th graders. Welcome to part 2 of this lesson in cells and classification. This is lesson 38, and we'll be covering pages 81 to 82. We will mostly talk about microscopes in this lesson. We talked earlier about living things and how living things have to have 5 characteristics. The first one, living things must grow and develop. When you were born, you did not stay a baby. Over time, you grew larger and larger and taller. But also, as you were growing, you were learning new skills, or you were developing. Number 2, living things can reproduce or make a copy of themselves. This is very important because all organisms grow old and eventually die, and we need new organisms to replace those organisms that have died. Number 3, living things respond to their environment. Now, you saw in the previous video how sunflowers move and follow the sun. The sun is a part of their environment, or non-living things. Number 4, living things use energy. You eat food every day, and that food that you eat, that's called digestion, along with the oxygen you take into your body, that's respiration. Those two things do a chemical reaction and make glucose, or sugar, that is used as energy for your body. This allows you to do work. Plants, however, they use photosynthesis to make sugar, so they can live and reproduce and grow and respond to their environment. Living things are made of cells, and whether it's a plant cell or whether it is an animal cell, all living things are made of cells. If it is something like your desk or your chair, you'll notice that it's not made of cells. Living things are made of cells. This man, his name is Robert Hooke. Now, he was a scientist, and he's not the inventor of the microscope, but he did make a more powerful microscope. And as he was observing things under his microscope, he was looking at a piece of cork. And as he was observing this cork, he saw small, very tiny chambers or rooms or spaces. He called them, those spaces, cells. Now, actually what he was looking at was dead cork, and he was just looking at the cell walls or the outside of a cell. It was empty. But if you have a living cell, it's filled with a watery substance called cytoplasm, which we'll talk about later in this chapter. However, today in modern science, a cell is actually the smallest unit of a living organism. For instance, the tiniest part of you that is an organism all by itself is called a cell. Now, a cell can be an organism all by itself, or it can be part of a larger organism working together. Now, this is some pictures. The first one is Robert Hooke's microscope. And then the second picture is what he observed under that microscope. This is that dead cork that we were talking about. This is what he saw. But the last picture on the far right, you'll see that is from a modern microscope, and it shows very clearly just the outside part, or the cell walls of this cork. And remember, dead things do not have anything in their cells. If it was a living organism, that cell would be filled with a watery substance called cytoplasm. But we'll talk more about that later. Now, these two gentlemen are from Germany. Their names are Matthias Schleiden and Theodor Schwann. They made 2 important discoveries in 1938. The first one, they realized that all living things were made of cells. I've kind of repeated this before, but this was their discovery, and they were following Robert Hooke's discoveries. The second one, that a cell can be an organism all by itself, or organisms can be a part of a larger organism. All of these discoveries that he made, the conclusions from their discoveries, are what we call cell theory. This man, his name is Zacharias Janssen. He is credited for the invention of the first microscope. He's a Dutch inventor. He made this microscope in the 1500s, but there's some argument whether or not he was truly the first person to invent the microscope. They believe his father, Hans Janssen, who was a lens maker, was credited with making the first microscope. But actually, his father began making what was called a compound microscope, which had many lenses inside of it to magnify an image. But it was actually his son Zacharias that finished his father's work. So they say that Zacharias was the inventor, but actually it was a combination of work of his father and Zacharias. This man developed microscopes further. He actually made a more powerful microscope than Zacharias. And in the 1600s or the 1660s, he discovered these little tiny creatures, and he named them animalcules. It comes from the word molecule. We learned about in another chapter. Animalcules, little tiny animals that he discovered in water. His microscope can magnify an image 300 times its original size. Now, in the 1930s, you'll see that this is the first electron microscope. And it was very, very powerful. It could magnify an organism or anything that is under the microscope 2,000 times its original size. But this is a more modern, or today's microscope, and modern electron microscopes can magnify images 500,000 times its original size or more. Now we're going to talk about the parts of a microscope. The first one is called a body tube. This is the very top of the microscope. Then we have what is called the nose piece. You can kind of think of the parts of a body to help you remember all these things. On the nose piece, you're going to have what we call objectives. And you have one that is a low power, medium power, and a high power. These three are what we call objectives. These are called stage clips. This holds the slide so an organism can be studied so it doesn't move on the stage. And then underneath, this is called the diaphragm. Under the diaphragm, you'll have a light source. Now, it will either be a light that you can turn on and off, or it will be a mirror that will reflect light through the slide so it can be seen clearly. Number 9, this is what we call the eyepiece. This is where you look inside. And this is what holds the objectives and the eyepiece in place. It's called the arm. This black area, this flat surface, is called the stage. And you have two of these. This is what we call a coarse adjustment, or the microscope moves. It moves very quickly up and down. But this next one, the fine adjustment, just moves the microscope just a little bit, just to get a perfect image of whatever they're looking at. And the last part is the base of the microscope. Now, you'll need to know all of these parts of the microscope, so please study this. You'll have a picture of this in your book, and also a picture of this kind of microscope in your activity manual. Okay, please read pages 81 to 82 and be ready with questions tomorrow, and I'll see you then.